All right. Good afternoon, everybody on the East Coast, and good morning to everybody who's in another other time zones in the country. My name is Rachel Liu. I'm a co-founder of Dashu, and welcome to the last virtual journal club of Dashu in 2019. So we have a very exciting talk today. Dr. Bo Huang from Pfizer will discuss the statistical consideration in the novel cancer research. And he's on now, so I'm very, very excited to hear his talk today. But before I hand it over to him, I would like to spend a few minutes to give you a brief introduction about our organization, Dashu, and also some upcoming exciting activities in 2020. So we are founded about 4.5 years ago, and we are a 501c3 nonprofit organization. We have right now, over 3,500 members around the globe. Our membership is totally free, and you probably got some newsletter or some event announcement from us from time to time. And you would know that we host a lot of different events. We organize international scientific conferences. We also have the monthly virtual journal clubs like this. We also collaborate with industry and also academia on the career development events, for example, those panel discussions to share tips and practical experiences in, when you get to a real job after school. We also have those educational lectures. For example, we have the legal series talking about IP protection in the past years. So we are a nonprofit organization totally run by volunteers. We are always actively seeking members, volunteers, and also new ideas. And you can visit us at our website at www.dashu.org. We have we share our upcoming events and also our past events notification and also archival materials on the social media. For example, we have the LinkedIn. We have actually two accounts. One is the company page. The other one is a user account. You can check us out and by searching Dashu Nonprofit or Dashu on LinkedIn. We also have Twitter. It's called Dashu Info is the handler. We have WeChat, which is a mostly Asian used virtual community. We also have a public account there. We always submit or publish our previous events on YouTube because we always record our virtual journal club and most of our technical seminars and post them on the YouTube channel. Our YouTube channel handler is also Dashu. For example, the November virtual journal club on 15 tips on making better use of our markdown by Yi Hui Xie has already published. And also in October, our prestigious Dr. Bin Yu talking about 3 principles of data science has also been published on our YouTube channel. So feel free to check us out. And we always welcome any suggestions and also comments to improve how we connect and also build our community. So one upcoming event that's co-sponsored by Dashu in 2020 is hosted by San Francisco Bay Area chapter of American Statistical Association, SFASA. That SFASA will have a holiday celebration on January 26th. It's on a Sunday. And from 2 o'clock to 8 o'clock in San Jose State University. So I think that is a in-person only event, but the keynote and panel discussions, as you can see, the speakers and panelists are really famous and also prominent in our field. Dr. Chito Hernandez from Biomarine. He's the group VP from that industry. And we also have Dr. Mukherjee from University of Michigan, who is also the chair of biostatistics there, and also Dr. Bin Yu, who is the Chandler's professor from Department of Statistics at UC Berkeley. So they will be our keynote speaker and also panelist for this holiday celebration. So come and join us, and Dashu is a proud co-sponsor for that event. About the virtual journal club, some of you have been participating for a while, and you probably would know that we started this series in September 2017, so that's already over 2 years, and we have hosted more than 20 monthly events and all archived on our website. So we invited world leader scientists to present their high impact research, and the material is free to you anytime. So looking into the new year 2020, again, we welcome speakers and topics, and your support by attending and spreading the word is highly appreciated. And anything, if you can think about that you want to chat with us, please email us. We have a Gmail account that's monitored regularly. And also we actively seeking sponsors for our virtual journals club. And in talking about the sponsor, so today we would like to thank our sponsor to this virtual journal club, StatForward. So StatForward is a specialized statistical consulting company who is specialized in biomarker and companion diagnostics and also clinical studies. So Dr. Shu Guanghuang would like to invite you to check out their website, which is listed on the slide. And also he welcome any questions if you have. So thank you very much, StatForward, to sponsor this virtual journal club for December. So without further ado, it is really my greatest pleasure to introduce you today's speaker, Dr. Bo Huang. So Dr. Bo Huang received his PhD in statistics from University of Wisconsin-Madison, and he joined Pfizer in 2008. Over the past 4+ years, he has been a director of biostatistics and oncology statistical group lead at Pfizer, currently leading 2 late-phase programs and 1 early-phase program. Dr. Huang has extensive experience in pharmaceutical industry across all phases of global clinical development of medical products, and he's also the recipient of the prestigious Craig Sexton Clinical Excellence Award at Pfizer in 2019. Dr. Huang is a recognized leader in developing and applying innovative quantitative methods, adaptive dose finding, and confirmatory designs in drug development. He's one of the main drivers behind the increased use of innovative model-based and adaptive designs at Pfizer. Dr. Huang authored more than 70 publications, so he published very extensively, including 50 papers and book chapters, and he published in top journals, including New England Journal of Medicine, Journal of Clinical Oncology, The Lancet, JAMA Oncology, Annals of Oncology, Biometrics, Statistics in Medicines, etc. His recent paper were also ranked by Wiley among the top downloaded paper in pharmaceutical statistics between 2017 and also 2019. He is also a US patent holder. He also received several poster and paper awards from American Statistical Association and also International Biometric Society. He is guest editor, associate editor, and also reviewer for scientific journals, and also an elected board director of the International Chinese Statistical Association. So without any further ado, I would like to hand it over to Dr. Bo Huang. So I'm going to stop sharing my slides, and Dr. Huang, please feel free to share on your end.
Can you hear me well?
Yes, we can hear you.
Okay, great. Perfect. So let me share my slide.
And also, while Dr. Huang is sharing his slides, just a housekeeping note, please mute yourself and also don't share your video. And if you have any questions to Dr. Huang, please enter your question in the group chat, and we will select the questions at the end of the presentation. All right, please, Dr. Huang.
Okay, thank you, Richelle, for the introduction, and thanks, Dashu, in particular, Sammy, for organizing this event and inviting me. I'm happy to be here. So statistical methodology in cancer drug research and development is a very big and broad area. There are a lot of interesting topics on different aspects of design, analysis, and interpretation of results. I had this privilege to work on oncology drug development for the past 12 years, so I would like to take this opportunity to share with you some statistical considerations that I think are both interesting and relevant to the novel therapies that have emerged in recent years. So first of all, a disclaimer. So this talk reflects the views of the speaker and should not be considered to represent views of the speaker's employer. In the US, the number of new cancer cases is expected to increase from about 1.5 million per year in 2010 to 1.9 million per year in 2020. Although the rate of people who got cancer is expected to stay about the same. And this is mainly because of population increase and higher rate of cancer screening and better screening techniques. So cancer can be detected at an earlier stage. And if you look at the plot on the right, the number of cancer deaths is expected to increase from about 575,000 per year in 2010 to 630,000 per year in 2020. However, the rate of people who die from cancer is expected to decrease from about 171 per 100,000 people in 2010 to 151 per 100,000 people in 2020. And this is also because of cancer being detected at an earlier stage and also the emergence of more effective cancer therapies, such as targeted therapies and immunotherapies. There are several types of cancer therapies. Surgery, radiation, and chemotherapy are the traditional cancer therapies. Hormone therapy is a treatment for cancer that uses hormone to grow, for example, breast cancer and prostate cancer. Stem cell transplants do not usually work against cancer directly. Instead, they help you recover your ability to produce stem cells after treatment with high dose of radiation and chemotherapy. But in multiple myeloma and some types of leukemia, the stem cell transplant may work against cancer directly. Targeted therapies and immunotherapies are the more novel cancer treatments that make cancer research really interesting and promising. Chemotherapies have been in use for several decades. Somebody's sharing video. However, and also chemotherapy still plays a very important role in treating patients with cancer. However, although they can kill cancer cells quickly, the drug distribution is not limited to the cancerous area. And healthy areas will be affected by chemo as well. As a result, general chemo has strong side effects and is not particularly successful as a therapy, and tumor cells often regrow. And as researchers have learned more about the differences in cancer cells that help them grow, they have been able to develop drugs that target these differences. And these drugs are often called targeted therapies. And targeted therapy is the foundation of precision medicine. For example, many targeted therapies go after the cancer cell's inner working, which is the programming that sets them apart from the normal healthy cells. And these drugs tend to have fewer side effects than chemo and are more effective and also enhance quality of life. There have been many targeted therapies being developed. However, many researchers believe that there's no better target in the human body than the immune system. And drugs developed that help your immune system fight cancer are called immunotherapies. So how does immunotherapy work? Cancer cells can bind to T cells and turn off their ability to detect and kill tumor cells. And immunotherapy drugs bind to T cells and keep them from being detected by blocking a tumor cell's access to the T cell. Based on the mechanism of action, current options for cancer immunotherapies include cancer vaccines, effector cell therapies, and T cell stimulating antibodies. And in solid tumors, the mainstream direction that has led to huge success is inhibitory antibody. Inhibitory antibodies are checkpoint inhibitors that block inhibitory receptors such as CTLA4 and PD-1. In other words, releasing a brake on the immune system. In contrast, stimulatory immunotherapies are agonist antibodies that target activating receptors. However, the results so far are mixed. In hematology or liquid tumors, CAR T therapies are very promising. CAR T therapy is a kind of cell therapy made by extracting some kind of T cells from the blood, genetically modifying them in the lab to intensify the immune system's response to cancer, and re-injecting them into the patient. Immunotherapy has been in the limelight over the past several years. Every year when people go to ASCO, immuno-oncology is at the center stage and grabs all the headlines and eyeballs. Even beyond oncology, immuno-oncology is also one of the most exciting research areas. For example, Science named immunotherapy as the breakthrough of the year in 2013. And most recently, the 2018 Nobel Prize in Physiology or Medicine was awarded to two oncologists for their pioneering work in immuno-oncology. So for statisticians, one challenge in IO dose-finding studies is to design a trial that manages late toxicities. Unlike chemotherapies that attack tumor cells directly, immunotherapies target the immune system to elicit effect on cancer cells. So this indirect effect can lead to late or cumulative effect on both safety and efficacy outcomes. And for safety evaluation, one concern is late onset toxicity beyond the traditional first cycle DLT observation window. And traditional dose finding designs based on the cycle 1 of treatment may fail to adequately assess the safety profile of the drug. Because dose finding trials are sequential in nature, having a multiple cycle DLT window will lead to very lengthy trial duration, which is not practically feasible. And some seminal work was done by Dr. Ken Cheng and Dr. Rick Chappell, who proposed the use of a time-to-event continuous reassessment method, the TICRM, which is a basic model-based design that evaluates time to toxicity instead of a binary variable of whether DLT occurs or not at the end of the DLT window. And this method can dramatically shorten the waiting period between those cohorts to make a long DLT window feasible. There have been a number of extensions to TICRM in the literature, including the so-called tight escalation with overdose control, the tight EWALK. In contrast to using a model-based approach, a rule-based design called rolling 6 design was also proposed as an extension of the traditional 3+3 design. The performance of this method is quite poor, just like its predecessor 3+3. And to take advantage of the simplicity of rolling 6, we are also incorporating the efficiency of model-based designs. Hybrid designs, also called model-assisted designs, were proposed very recently, including the tight Boeing and the POD TPI designs, which are extensions of the Boeing design and MTPI design. The POD TPI design is not published yet, but will appear soon in a special issue I'm currently editing for Statistics in Bioscience. Both the model-based designs and the hybrid designs are very useful in my opinion and should be used more often in clinical practice. So here is a phase 1b oncology study evaluating the safety of combining two immunotherapies, utomilumab, a 4-1BB agonist, and pembrolizumab, a PD-1 checkpoint inhibitor. This study, the results of this study were published in Clinical Cancer Research in 2017, utilizing a modified TICRM design with cyclical adaptive weight function based on a paper I wrote a few years ago for Biomedical Journal. By using this novel design, the clinical trial duration was substantially shortened, and dose escalation was completed in 6 months, although a 2-month DLT window was considered. Another challenge in IO dose finding these studies is the optimization of the recommended phase 2 dose. Although it is true that higher dose will likely lead to higher toxicity, it is not necessarily the case for efficacy. And higher dose may not be associated with higher activity due to, say, for example, overstimulation of the immune system. The same challenge also present for dosing schedule. And there are novel methods published in the literature looking at composite endpoints of safety and efficacy. And there's also a conference paper authored by Richard Pasteur from FDA on this topic. However, despite the great efforts by statisticians, the use in practice is very limited. There are several reasons behind this. First of all, these designs are complex model-based designs with a number of parameters to be estimated. Well, we know phase 1 trials are very small, and statistical software with GUI capability need to be available. Second, efficacy requires much longer follow-up than safety, so a time-to-event approach may be necessary. In addition, phase 1 trials are often all-comer studies. Unlike safety, efficacy is very sensitive to patient heterogeneity and could shift dramatically in different populations. A common practice in clinical trials is the implementation of a pragmatic approach. First, do a conventional dose-finding study for safety only, and then conduct adequate retrospective analysis of PK biomarkers and investigate the dose exposure relationship and assess early efficacy signal to have a good understanding of the data. And in the third step, evaluate 1 to 3 doses in the expansion cohort in a homogeneous patient population and randomize patients to the selected dose groups. Another challenge in IO dose finding study is to identify the dose for drug combinations. In the field of immuno-oncology, it is believed that combining an immunotherapy with another IO drug or other drugs of different mechanism of action, such as chemos and targeted therapies, can elicit synergistic effect and significantly boost efficacy. So 1 plus 1 equal to 2 or more than 2. However, the complexity of the phase 1 trial design increases exponentially with the number of different drugs included in the combination strategy. Further, unlike single-agent dose escalation, where monotonicity is generally assumed to be true, for dose escalation for combination agents, only partial ordering is known for the dose toxicity relationship. So there are a number of methods on how to tackle those finding combinations. For example, we can consider one-dimensional escalation, such as alternate escalation of the agents. 2, simultaneous escalation or diagonal escalation of both agents. And 3, escalation of one drug to the recommended dose for the phase 2 trials while holding the other agent at fixed dose. However, these are simplified approaches that may not be optimal. And, and in the case of a bidimensional dose escalation, flexible model-based designs were also developed with model parameters to account for the inherent complexity of drug combination in the dose toxicity relationship. And we can build an MTD contour instead of a single estimated MTD. So for efficacy evaluation of immunotherapies, predictive biomarkers can play an important role. So what is a predictive biomarker? A predictive biomarker is different from a prognostic biomarker. A prognostic biomarker provides information about the patient's overall cancer outcome, regardless of therapy. Well, a predictive biomarker gives information about the effect of a therapeutic intervention. Predictive biomarkers are critical to development of targeted therapies, and which is the foundation of precision medicine. and have played an important role for checkpoint inhibitors as well. Ignoring the effect of a predicted biomarker can lead to costly consequences, such as a trial failure. However, dichotomizing patient population as marker positive population versus marker negative population based on predicted biomarkers may not always be straightforward. For example, for checkpoint inhibitors, two notable biomarkers, PD-L1 expression and tumor mutational burden, are not perfect biomarkers because they are continuous variables. It is different from some biomarkers for targeted therapies based on genetic mutation or translocation, for example, EGFR. And also the various companion diagnostics used by different companies make biomarker testing challenging and expensive. So with the increasing use of next generation sequencing, it is believed by many people that genetic testing will be more standardized and cost effective. In recent years, efficient biomarker-based designs and master protocols have been advocated in medical field, in particular for oncology and immuno-oncology. A master protocol is a single overarching protocol designed to study multiple patient populations, multiple diseases, multiple therapies, or all of this. There are three types of master protocols: basket trial, umbrella trial, and platform trial. A basket trial studies the activity of a single therapy or combination of study drugs or other therapeutic interventions in the context of multiple diseases of disease subtypes. An umbrella trial studies the activity of multiple therapies in the context of a single disease that can include multiple disease subtypes and therapies specific to the disease subtypes. And the platform trial studies multiple therapies in the context of a single disease or multiple disease subtypes in a perpetual manner, where therapies may be added or dropped from the platform on the basis of a pre-specified decision algorithm. So this is definitely an exciting era in cancer research to develop precision medicine. And in 2018 alone, there were 19 cancer drugs approved by the FDA, and many of them are precision medicines. So other challenges and probably the most relevant issues in immuno-oncology efficacy evaluations are non-proportional hazard, selection of intermediate endpoints, and to quantify treatment benefit more accurately. And this part is also the focus of my talk. In late phase development of immunotherapies, one challenge is that the proportional hazard assumption, commonly assumed for time to event endpoints, may no longer hold. It is quite common to have late separation in curves due to delayed anti-cancer effect, especially for single-agent checkpoint inhibitors. The responses induced by immunotherapies are usually much more durable than traditional therapies, which can lead to long-term benefit, and some people call it a cure effect. So when there is a late effect and a long-term effect, we need to ask ourselves whether the gold standard combination of log-rank test and hazard ratio is still optimal, and how do we design the trial with interim analysis, in particular with a futility boundary. Secondly, there could be a weak or even negative correlation between short-term endpoint, progression-free survival, and long-term survival endpoint. So we need to be very careful when designing a futility interim analysis based on PFS. And perhaps PFS is not a good intermediate endpoint in immuno-oncology studies. And lastly, in the presence of non-proportional hazards, how do we quantify or estimate treatment benefit? The statistics field has been obsessed with p-values because we need a significant p-value to get a drug approved. No question about that. But is it really ultimately? The p-value itself has no clinical meaning at all, and there are so many caveats associated with it. And there is a special issue in the Journal of the American Statistician, so I don't have to spend time talking about this. For survival analysis in clinical trials, one key component is the scientific choice of a summary measure, which is not only essential to the estimate framework, but also the foundation of making statistical inference. In randomized comparative studies, the summary measure can be based on horizontal separation of the curves, which is the percentile of survival function. The median is the most common percentile and has been used ubiquitously in oncology presentations and publications. The problem with this approach is that there are infinite number of choices, so it is not a global summary statistics. Plus, the median is unstable, and 95% CI is usually wide. And likewise, we can also look at the vertical separation of the curves, which is the event-free probability at a specific time, such as 1-year survival rate, 5-year survival rate. However, just like the percentiles, there are infinite number of choices, and these are not global summary statistics. Alternatively, we can look at the slope of the curves at any particular time point relative to the survival probability at the time point. And statistically, that gives us the hazard rate. To evaluate treatment benefit, we can look at the relative hazard of two arms, which is the hazard ratio. However, the hazard ratio is a function of time. So in order to come up with a single metric, conventionally, the hazard ratio is averaged over time with pre-selected weights. And under the semi-parametric proportional hazard model, the locally optimal weight leads to the log-rank test. And optimality under the proportional hazard assumption also contribute to the popularity of the log-rank test. Although the proportional hazard assumption largely holds in the era of cytotoxic drugs, it is often violated in immuno-oncology studies. So given the indirect effect of immunotherapies on cancer cells, it is not uncommon to see no separation of curves in the first several months before the treatment activity kicks in. And the tail of the IO curve is often flat with few events thanks to some patients having a durable response to the drug. This non-proportional hazard pattern is quite different from what we typically see in targeted therapies, where there is usually a quick and substantial effect early on, but the effect fades away over time because of mutation of the cancer cells that can evade the attack of the drug. So despite some very nice theoretical properties, the log-rank test and hazard ratio come with limitations, especially under NPH. As I said earlier, the log-rank test is optimal under the proportional hazard assumption, but may lose power when the assumption does not hold. And we know the power of log-rank test is event-driven. So with small events, then the power is low. And when we have flat tails, meaning there are few events with long-term follow-up, then the log-rank test is not sensitive to this kind of effect. Also, importantly, censoring can also introduce bias under NPH. So we have two studies with identical Kaplan-Meier curves, but because of the different distribution of censoring, we may end up with different hazard ratios from Cox model. The interpretation of the hazard ratio from the Cox model is also problematic under NPH, and we know it is only a relative measure of effect. Given these limitations, there have been a number of alternative methods proposed in the literature, just to name a few. The weighted log-rank test assigns an equal weight to events occurring at different times, such as the Wilcoxon test, the Fleming-Harrington PQ family test. So for example, if there is a late effect of an IO drug, by assigning more weight to later events, the weighted log-rank test will have more power than the log-rank test. However, oftentimes, we do not know if there's an early effect or if there's a late effect. So if the assumption is wrong, a weighted log-rank test can actually lose power. As a result, some researchers propose versatile tests or adaptive tests where the weight is pre-specified to be determined by the data, but in an agnostic way at the design stage to minimize bias. For example, a Young and Prentice for rank-based test, and Uno and colleagues for the Kaplan-Meier-based test. Alternatively, we can also use the so-called maximum combination test which is now shown on the slide. But the idea is to choose the maximum test statistic from a number of weighted log-rank tests. That is the test recommended by the cross-farmer NPH working group and presented last year at the Doug Margoli's workshop. And we just had a paper published in Statistics in Biopharmaceutical Research this month. The win ratio method is another very interesting method, which is a generalized pairwise comparison method by looking at the ratio of win probability in each arm. And the net benefit method is a similar method by looking at the difference of win probability. And it is a non-parametric method that has intuitive interpretation. And I will go over this method in more details in the next several slides. And last but not least, the Kaplan-Meier-based method. The Kaplan-Meier-based method, in particular, the restricted mean survival time, the RMST, has garnered a lot of interest and attention in the statistics field and also in the medical field because of its appealing clinical interpretation and the ability to measure long-term treatment benefit. So which method should we choose? All methods have pros and cons. The versatile test or adaptive test is attractive because it provides robust power in all situations. However, for me personally, I would prefer a method that can not only address the power issue, but also have a coherent target estimate that is clinically meaningful. And I always think estimation is as important, if not more important, than the p-value. Although we do need a significant p-value to get a drug approved. Because of this, I really like the win ratio method and the RMST method, and I will go over them in details later. The win ratio method. The win ratio method is a generalized comparison method. It compares the outcomes of each patient in the treatment arm with each patient in the control arm with 3 results: win for the treatment, win for the control, and a tie. A tie often happens in sensor data because a win cannot be determined. So this concept is very straightforward and intuitive. And the win ratio equals the ratio of the win probability in the treatment group to the win probability in the control group. So when the proportional hazard assumption holds, the win ratio is equivalent to the inverse of the hazard ratio. For the observed win ratio, if we use a non-parametric counting approach, it strictly follows the counting process with some very nice theoretical properties. The win ratio method is particularly useful to analyze semi-competing risk data when there are non-fatal events and fatal events. The win ratio method can prioritize multiple outcomes. When usually the fatal event is more important. So for example, if there's a tie for the fatal event, then the non-fatal event is further evaluated. This is particularly appealing because it uses more information than, say, a composite endpoint of time to first event, such as progression-free survival, and can increase power. It also ranks the events by order of importance. This method is recommended if the following 2 conditions are satisfied. The more important endpoints tend to occur later and are reasonably frequent. Also, censoring is not substantial because censoring can have a big impact on the win ratio results. And we have a paper just published last month on the impact of censoring and follow-up time of win ratio, and also using the win ratio to handle non-proportional hazards. The win ratio method for prioritized multiple outcomes has been applied in rare disease. For example, a study investigating tafamidis in TAC was published last year in the New England Journal of Medicine. And in the primary analysis, they hierarchically assess all-cause mortality followed by frequency of cardiovascular-related hospitalizations, according to the Finkelstein-Schoenfeld method. And the win ratio statistic was used to summarize the treatment effect and to interpret the test result. The next method I want to really focus on is the RMST. In recent years, in the medical field and the statistical field, people are increasingly interested in the area under the survival curve as a summary measure, which is basically the average survival time up to a specific time point. The RMST is a method originally proposed a few decades ago, but it hasn't gained much attention until in recent years. The RMST has an intuitive and clinically meaningful interpretation. If we use the Kaplan-Meier estimator for the survival function, then the RMST estimator is non-parametric, and statistical inference can be done on the observed data based on some existing work in the literature. Compared to the hazard ratio, the RMST has a very good clinical interpretation, regardless of whether the proportional hazard assumption holds or not. And if we use the Kaplan-Meier estimator to estimate RMST, the RMST method is non-parametric. Also, the RMST difference and the RMST ratio provide dual presentation of both absolute and relative effect. And compared to the median and t-time event-free probability, the RMST is an informative global summary, not based on arbitrary percentile or cutoff. And because the RMST depends on the cutoff point, tau, we can also look at the RMST curve, which provides a temporal profile by looking at different transition points. And if you look at the plot on the right as an example, so this is a phase 3 trial of inotuzumab versus standard chemotherapy in acute lymphoblastic leukemia. The blue curve is the treatment arm, and the red curve is control arm. And if you look at the plot, it is quite apparent that the proportional hazard assumption does not hold. And the top plot shows an OS median difference of 1 month and a 12-month OS rate difference of 4%. The one-sided p-value for log-rank test is 0.02. And if you look at the bottom plot, the OS mean difference is almost 4 months, which is the area between the curves, with a one-sided p-value of 0.002. So most people would agree that in this specific example, the area between the Kaplan-Meier curves is a better summary of the treatment effect. In the past, I heard criticism against the RMST method on the requirement of a truncation cutoff point. Some people say it is a limitation of the method. It is a little bit ironic since people really make a case about this because the name includes the word restricted. But not many people care about the fact that the same requirement is also applicable to other summary statistics based on the Kaplan-Meier plots, including the hazard ratio and the log-rank test, which is dependent on the follow-up time. As a matter of fact, the maximum allowed time window for hazard ratio and log-rank test is even narrower than that for the RMST. And under some mild conditions on the censoring distribution, one can make inference on RMST up to the last follow-up time, either event or censored, for the arm with shorter follow-up time. In contrast, for the hazard ratio and log-rank test, one can only use data up to the minimum of the last event time for any arm, the last follow-up time of the arm with the shorter follow-up. So looking at the same example from the previous slide, the window for hazard ratio and log-rank test is up to a little more than 30 months, while the window for RMST is up to more than 37 months. So the RMST uses more information from the observed data. And there are two references. The RMST is getting much attraction in the oncology field in recent years, and there were quite a number of papers published in top-tier journals, top-tier medical journals, just to list a few. Historically, RMST has been used as a supportive analysis method, but most studies were designed based on the hazard ratio and the log-rank test. So people may ask, what if we want to design a trial using the RMST as the primary method and do sample size calculation based on the RMST. So in order to do that, the first step is to construct a hypothesis test. Assume we are interested in the difference in the RMST between the treatment arm and the control arm at truncation time tau. The null hypothesis can be built as the difference less than or equal to 0 versus an alternative, the difference greater than 0. And we are interested in detecting a difference of delta to achieve a desirable level of alpha. In the literature, there are 3 papers published on the design of RMST and sample size calculation. Peck and colleagues propose a simulation-based empirical approach. Under some parametric assumptions, for any sample size and fixed follow-up time, one can simulate the power. So by doing a grid search, one can find the minimum number of patients in order to achieve a desirable level of power. Royston and Palmer provided a calculation of the RMST and its variance based on the combination of a simulation-based approach and analytical-based approach by using piecewise exponential distribution for event distribution and simulation approach for censoring to properly inflate the variance. To implement a fully analytic approach, we recently proposed a statistical framework based on parametric assumptions for event, censoring, and accrual. And the work was published this year in the Journal of Clinical Trials. So I'm going to skip the next several slides in the interest of time, because I do want to leave some time for another very important topic. This one, skip. Okay. For the next part of my talk, I would like to introduce a method that is an extension of RMST to multi-state survival analysis, a new intermediate endpoint to measure the effect of cancer treatment, including immunotherapies. As cancer is becoming a chronic disease for the major indications, for clinical researchers and drug developers, we need to ask ourselves, what does a desirable future cancer treatment look like? A future cancer treatment may not be a cure, but can effectively control the disease, and patients can live a normal life with the disease for a long period of time. So what are the characteristics of a desirable cancer treatment to patients and doctors? First of all, it needs to be life-extending, and OS is always the gold standard. Second, there should be a high likelihood of tumor response, defined as reduction in tumor size. No patients want to carry a large burden of tumors and live a miserable life. Also, once receiving the drug, time to response should be fast, and the amount of time being in a stage of tumor response should be long. That is, the response should be durable. So these 3 are the so-called imaging-based endpoints. Other desirable characteristics should include manageable side effects and improved or not worsening health-related quality of life, as patient-reported outcomes are becoming more and more important. A composite endpoint is proposed that can effectively measure the 3 desirable outcomes based on tumor imaging. So a natural question people will ask is, wait a second, for an imaging endpoint, we already have PFS, right? Well, although PFS is a widely accepted intermediate endpoint, sometimes it has limitations. As we know, PFS measures the duration of disease stabilization, but disease stabilization may not translate to long-term survival benefit. It can also not distinguish a tumor reduction from no change or slight increase. So if you look at the example on the right, patient A and patient B both had PFS of 12 months from treatment initiation. However, patient A's target lesions had slight increase most of the time during the course. In contrast, patient B had dramatic reduction in tumor burden and achieved complete response after 6 months. The patient had a target lesion in the lymph node So the lymph node lesion reduced to normal size. And after a few months, the lymph node lesion was enlarged and the patient had progressive disease. So both patients had the same PFS of 1 year. But from a clinical perspective, patient B had a much better outcome during this period. And lastly, although PFS enabled us to design smaller and faster trials in the past, as cancer becoming a chronic disease, we will lose this capability. So I would like to share with you an interesting commentary article that was published in The Lancet in May this year. So the authors raise a thought-provoking question. Are modern clinical trials using the right endpoints to be truly pivotal? If the desired goal is to increase the proportion of patients achieving a durable complete response, perhaps it is time to formally incorporate this endpoint in registrational studies and to consider a durable complete response as a surrogate for overall survival in this increasingly complex therapeutic landscape. So indeed, as cancer is becoming a chronic disease, improving OS in clinical trial setting is going to be very difficult with multiple lines of effective treatment. The same thing is for PFS. As PFS is getting longer and longer, response-based endpoints, including response and duration response, have the potential to play a more important role in future oncology studies. The traditional way to analyze duration response is to analyze duration among responders only in an empirical way. Although DOR among responders have clinically meaningful interpretation, we cannot make inference, statistical inference, comparing two arms because response is an outcome after randomization. And responders in different populations can have different prognosis. For example, patients may be more likely to respond to chemo due to better conditions and prognosis. So it's not an apple-to-apple comparison. And if the ORRs are different, then we may have an interpretation issue. So for example, if you look at the plot on the slide based on a real phase 3 study, this shows the Kaplan-Meier plot of duration response among responders for the experimental arm in cyan and versus control arm in red. The responses are durable in both arms with median not reached. However, the number of responders in the experimental arm is almost double that in the control arm. So comparing the DOR among responders can be quite misleading. Alternatively, we can use the Kaplan-Meier approach for all patients by assigning DOR equal to 0 for non-responders. The initial probability of DOR greater than or equal to 0 reflect the objective response rate. The plot on this slide shows that the curves start at 51% for the treatment arm and 26% for the control arm, the respective response rate in each arm. However, there's a fundamental issue with this approach. The Kaplan-Meier approach is not valid due to dependent censoring. This is because both DOR and this censoring time are measured from the time of the initial response, which itself is an outcome of treatment. So censoring time is not independent. If time to response is short and duration of response is long, the impact is small. But no matter what, censoring is no longer independent, making this naive approach biased. And from a clinical perspective, if the ORR is less than 50%, then the commonly used metric median is equal to 0, which has no meaning and cannot be used to quantify the treatment effect. So this is a recent paper published in JAMA Oncology, and we propose an intuitive composite endpoint of duration response in the ITT population, not just for the responders. And it effectively addresses the dependent censoring issue associated with the Kaplan-Meier approach. As a result, it allows us to do statistical inference between arms with good clinical interpretation. The concept is very simple and intuitive by looking at 3 events of interest: response, progression, or death, where response is a good event, while progression and death are bad events. So to evaluate treatment effect of the drug, there are 3 states. State 0, from time 0, either randomization date or first dose date. State 1, from time 0 to response, progression, or death, whichever is earlier. And state 2, from time from response or stage 1 to progression or death. An effective treatment should keep patients in stage 2 for as long as possible. So the parameter of interest is the mean duration of a patient expected to spend at stage 2. And for responders, this is equivalent to the duration response by traditional definition. And what we have proposed is a composite endpoint by taking into account TTR, time to response, response rate, and duration response altogether in the ITT population by considering DOR as a multi-state statistical problem. In multi-state survival analysis, we can take advantage of the nice property of the mean that the expectation of variable A minus variable B is equal to expectation of A minus expectation of B. And assume some patients are still in follow-up, we need to select a common cutoff point for tau for this analysis. Then the mean duration response is the expectation of time to progression or death minus time to response, progression, or death, whichever is earlier. And it is equal to the area under the curve of PFS curve minus the area under the curve of progression, death, response-free curve. So it is the area between the PFS curve and the PDR event-free curve. It can be interpreted as the expected duration of response for a patient receiving treatment. So this is a very intuitive interpretation. And this is something a physician can tell his patient before the patient takes the drug. The recommended cutoff point tau is the minimum of the largest PDR event-free time for either arm. And it is the latest time point we can choose. Since the mean duration response is the area between the two Kaplan-Meier curves, and the two curves are correlated, the calculation of variance is a little bit tricky. And it can be derived either by bootstrapping or analytically based on counting process theory. The R code is available to implement this method and can be downloaded on Professor Lu Tian's website. And here is a graphical example of a restricted mean duration response. And this is a tweet by JAMA Oncology posted on Twitter. I'm not going to interpret the result in details in the interest of time. So the RMST method has been implemented to support the submission, the EMA submission of avelumab plus axitinib in first-line renal cell carcinoma. And it was well received by CHMP and the rapporteur. It was also presented in an oral presentation at ASCO GU this year and used to support two poster presentations at ESMO this year. And recently, there was a paper published in JCO in September. The authors proposed a novel outcome measure, treatment-free survival, using the same method as in our duration response paper. So in summary, extending RMST in multi-state survival analysis is an interesting topic that warrants further research. So immuno-oncology is an exciting research area that may offer a cure to patients. And conventional gold standard may not be optimal. And this poses challenges and also opportunities to statisticians to develop and implement most effective quantitative methods. And I believe the time spent will be paid back with enhanced clinical trial design, analysis, cost saving, and improved probability of technical and regulatory success in the long run. And I would like to end my talk with a racing car analogy. I don't know how many of you are electric car fans, so my apologies if you are a fan of a gasoline car. On the top left of the picture, we have a vintage racing car in the early 20th century. And on the bottom right, we have a 2020 Tesla Roadster, the fastest car on Earth, 0 to 60 miles per hour in only 1.6 seconds. So this is a lot like the evolution of cancer treatment in the past century. And we are in a very exciting era with breakthroughs in biotechnology and innovative cancer treatments such as targeted therapies, immunotherapies, cell therapies, gene editing therapies, just to name a few. And I'm optimistic that statisticians and data scientists will thrive and play an important role. And lastly, I would like to acknowledge my collaborators, Professor L.J. Wei, Professor Lu Tian, Dr. Xiaodong Luo, Dr. Gao Hongdong, who have made significant contributions to the materials that I presented. I would also like to thank a few Pfizer colleagues for sharing the data of their studies, which I use as examples. So thank you all for your attention. This is the end of my presentation.
Thank you so very much, Dr. Huang. This is really inspiring, and also, thank you so much for your excellent presentation. We actually have some questions that we received from the chat. So there are two questions for you, Dr. Huang. One is about the win ratio statistics, and the other one is about FDA's view or acceptability. So for the first question about the win ratio statistics, Mike asked the question, does a win mean that time to event is longer? And also, how does the win ratio related to the C statistics? So that's our first question.
So that's a very good question. So the win or lose is something that should be defined by study-specific endpoints and pre-specified in the protocol or the SAP. The win ratio method can be implemented not just for time to event endpoint, but also can be used for binary endpoints and continuous endpoints. And for binary endpoints, it is actually equivalent to the stratified odds ratio, something like, if you use the stratified win ratio, it is equivalent to the Cochrane mental hazard statistic. And for time to event endpoint, because of censoring, and if we have multiple prioritized endpoints, there may be different situations. Let's consider the simple situation that we just look at one endpoint. And if that's the case, a win for the treatment means that the time to event is longer than the time to event for the control. And if there's censoring, for example, if there's censoring in the treatment arm, then the time to censoring should also be longer than the time to event for the control arm. So that's a win. If it's in the opposite direction, let's say we have a censoring in the control, and there's no matter what result in the treatment arm. So if the time to censoring for the treatment is longer than the time censoring in the control, this is a tie because of censoring, we cannot tell if there is a win for the treatment or a win for the control. So at the protocol stage, it should be pre-specified very clearly because there are different ways of defining win or lose, really depending on the specific situation and the endpoints. And the win ratio statistic is actually commonly related to a number of statistics. For time to event endpoint, if the proportion hazard assumption holds, it is equivalent to the inverse of the hazard ratio. And for binary endpoint, it is equivalent to the Cochrane. If we use it is equivalent to the odds ratio. And if we use the stratified win ratio, it is equivalent to the Cochrane mental hazard statistic.
Okay, thank you. The second question from Snow is, what is FDA's view or acceptability of pivotal trials with trial design and analyses based on RMST?
So that's another very good question. So there was a cross-farmer FDA industry workshop on non-proportional hazard, and this working group was established, I believe, like 3 years ago. And we have evaluated multiple methods. And by the way, this workshop was initiated by the FDA because they also are well aware of this non-proportional hazard issue and the impact on the study results, not only on the power but also on the interpretation. And there was some recommendation from the working group for the testing part. The recommendation is the maximum combination test. And for the estimation part, there are a number of recommendations, including the restricted mean survival time, because it has really clinically meaningful interpretation, and it doesn't really rely on the proportional hazard assumption. The acceptance of the FDA, I think this depends on how it's being used. If it's being used as a supportive analysis, the FDA has no issue with that. But if it is being used as a primary analysis, the FDA may be requiring further evaluation in clinical trials and to gain more data. So there's no clear answer to this because I'm not from the FDA and I cannot speak for the FDA. And they will always just say, it's a review issue, right? But so far, RMST has been used particularly in the estimation of treatment effect when the proportion hazard assumption does not hold. So in terms of using it to design a study, the methodology is being developed and has been developed, like the method I just shared about the paper published in clinical trials on the design and monitoring of clinical trials using RMSD. So these are new methods, and over time, with more people using it, it's more likely for FDA to accept it. Even for the adaptive design, many years ago, FDA issued a guidance document on adaptive designs, and they call some designs well understood designs, and some designs not so well understood designs. That was many years ago, and since then, because of the increasing use of adaptive designs in clinical trials, some of the not so well understood designs may have become well understood designs. I think this is the same case for RMST and also some other methods. So it really requires clinical trial practitioners to use them in clinical trials, have them published, gather more data and experience, so that FDA and also other health authorities will become more comfortable with the new approaches.
Mm-hmm. I thank you for this excellent comments. For any new methodology in research and also, people develop it, it always takes time for FDA to implement or consider or even consider in the regulatory approval pathway. So that sounds like something that, we in industry and also academia needs to work with FDA in the future to find a good way to benefit the patient in the end. So thank you so very much, Dr. Bo Huang. Thank you so much for your insightful presentation. And in the interest of the time, I would like to conclude this virtual journal club. Thank you so very much for everyone participated online and also for your excellent questions and also for Dr. Huang's very helpful answers. And I would also like to thank all the volunteers from DASHU. Without you, this wouldn't happen, and also the entire series wouldn't happen. And also thank you for our sponsor, Step Forward, for sponsoring today's event. With that, I would like to thank you all and also happy holidays to everybody.
Happy holidays. And thank you for having me.
Thank you.
Bye-bye.